Microfluidic devices used to compartmentalize neurons have become a standard tool in neuroscience, enabling researchers to physically and chemically manipulate subcellular regions of neurons, including somata, axons, dendrites, and synapses. These compartmentalized culture devices can help answer numerous questions in the neuroscience field, such as how axons extend during development to form synapses, how synapses remodel and reform following axon damage, and how pathological conditions can transsynaptically propagate in diseases such as Alzheimer's.
This protocol describes the use of pre-assembled plastic multi-compartment chip for compartmentalizing cultured primary rat neurons. The main advantage of using these chips is that they provide an easy-to-use and highly reproducible method to compartmentalize neurons.
Another advantage is that these chips yield healthier long-term growth of neurons and isolated axons than previous silicon-based compartmentalized devices, and are equally compatible with high-resolution microscopy.
Other model systems, including human stem cells, are also able to be cultured within the pre-assembled compartmentalized chip.
To begin, place a sterile multi-compartment chip into a petri dish. Add 100 microliters of a pre-coating solution to the upper left well of the chip and allow it to flow through the main channel into the adjoining well. Next, fill the lower left well of the chip with 100 microliters of the pre-coating solution. This time, wait 5 minutes to allow the solution to flow through the microgrooves. Now, add 100 microliters of the pre-coating solution to